Good morning, everyone. This is a new product we're launching here. It's called the QTRam this year at CPhI North America. The QTRam stands for Quantitative Transmission Raman Analyzer, and it's basically targeted around the content uniformity and blend uniformity applications. We're in booth 1612, so feel free to stop by for those of you who are interested in getting a closer look at the system. I'll be touching on a few things today, some applications or a little bit more detail about content and blend uniformity, current analytical methods that are used for this application, advantages of our technique, which is the transmission Raman, and then going a little bit more into detail of the QTRam and ending with a application example. So to begin with the applications, there are a number of unit operations in the pharmaceutical process for oral solid dosages. It starts with raw materials processing and dispensing, milling, wet granulation, sizing, and so we're targeting on blending and tableting. Those are the two unit operations that we're targeting for this product. So blend uniformity is actually the adequacy of mixing to assure that uniformity and homogeneity of the finished product is achieved. And content is a mandated test by all the regulatory bodies to make sure that your unit drug dosages are within a very specific label claim criteria. You can also use this system for formulation development to actually quantitatively analyze the excipients and active ingredients in your tablets or capsules, and also for counterfeit detection. So these are the four main uses or applications of the QTRam system. The USP and the EP both touch on chapters. There's 905 and 2.9.40 in the European Pharmacopoeia. And it's actually, again, like I said, the test to verify the consistency of the drug substance that each dosage unit within a batch is actually within a narrow range of your label claim. Usually it's 75% to 125% is the variation allowed from the label claim, 100% being the target dosage. So there are certain criteria that people follow for blend uniformity. You actually take 3 replicate samples from at least 10 locations in your blender or reactor, and then you assay 1 sample per location. Then there's an acceptance criteria like you can see up here. And if it passes, then your test is passed. Otherwise, you move on to additional testing. Similarly, for content uniformity, you take at least 1 dosage unit from 30 different locations equally over your batch. Then you assay a total of these 10 units, and then 75% to 125% is the criteria usually applied for this test. And if it passes, then you can release the batch. Some of the current analytical techniques or methods used and the advantages of transmission Raman spectroscopy. So currently a lot of the industry focuses on using HPLC, which is a very popular technique. It's a lab-based technique. The sample preparation, some of the disadvantages, the sample preparation required for this technique is a lot. It also involves solvents, and it's a destructive technique. It has a very long analysis time, especially for tablets that can be longer extended release or stuff like that. And also it cannot tell you other properties of the substance like crystallinity, polymorphism, etc. NIR is also a technology that is sometimes used for this test. And it's not as specific as Raman spectroscopy, and the moisture and particle size can also impact the readings that you get from this. So Raman spectroscopy is based on light scattering. Essentially, you excite the molecule with a monochromatic source of light, a laser, and you read the inelastic scattering of light. And in this case, it's transmission based, so you read it from the other side of your sample. So the light actually travels or traverses through the entire body of the sample. So this is the actual difference between conventional Raman spectroscopy and transmission. In conventional backscattering Raman, you excite the sample from one side and you actually read off the backscattering. So you only tend to observe the molecules that are near to the surface, at the surface, or just a little bit below the surface. In transmission, you're actually exciting the molecule from one side and collecting the Raman or the inelastic scattering from the other side. So your laser is actually traveling through the entire sample or volume of the tablet and giving you a more volumetric content measurement or averaged measurement. Even in the USP Chapter 1039 chemometrics, they mention specifically NIR and transmission Raman as the two techniques that can be used as alternatives to HPLC for content uniformity measurements. And it clearly states some of the disadvantages as well. So from that perspective, there is a strong reference in these pharmacopoeias for using transmission Raman for content uniformity as well. Some of the benefits now of transmission Raman is that it's rapid and non-destructive, so you can actually store the batch of samples that you tested with Raman, whereas in HPLC you destroy the entire batch. There's very high chemical specificity, so the peaks that Raman spectroscopy usually shows are very narrow and they're very specific to the molecule itself. Raman can also provide structural information, like you can tell you differences between the polymorphic states and also crystallinity. You also sample a very large volume, like 3, 4, or 5 millimeters of a spot size. That means that you can actually tell an average spectrum of the entire reading of your tablet or capsule. There's absolutely no sample preparation required, so you literally pop the tablet onto the sample stage and take a 20-second scan to tell you exactly what the content is, and no consumables. So you save on a lot of those costs as well, switching over to transmission Raman spectroscopy. A basic workflow for content uniformity is that you first, for Raman spectroscopy to be used for content uniformity, is you first do some exploratory work and you find out where your API peaks lie or your active peaks lie within an average spectrum of API and excipients. You prepare a calibration set of samples which go from basically 70% to 130% of your label claim, and then you collect a separate validation spectra and then you validate this method. Basically, the model that's used all the time is PLS1, partial least squares, and then you release this method to be used in your daily operation. So that's a basic workflow around the content uniformity application. So let's touch a little bit about on what our system is, the QTRam. It's actually built on our Pittcon Gold award-winning high throughput Raman platform. The spectrometer actually can be, we can modify the laser spot sizes from around 2 millimeters up to 8 millimeters, so you can really get a uniform reading or a large average spectrum of your content. It has a special transmission module, which you can see at the side there in the white. It's called the QT sampler. And we have a fully 21 CFR Part 11 compliant software called the BW Analyst. And this can help you build the methods that you require, validate them, and also do routine analysis. We also offer a chemometric software called BWIQ, where you can build these PLS methods. And like I said, we can actually modify the spot sizes and also change or modify or customize for you the different sample holders to match the shape and sizes of your different tablets. The software, I'm just going to show you a couple of screenshots of the software. It actually has 4 or 5 different tabs to cover the entire operation, so you can actually build a method in the orange tab. You can do daily operation in the blue one. There are a whole lot of settings, and reports can be printed out as well directly, and you can run daily performance tests as well. It actually guides you through the whole process, or your developers through the entire process of creating a method and validating it. So you collect a bunch of spectra, you export them to be able to build the method in BWIQ. You build it, then you re-import the model that you build, and then you validate, collecting a separate validation spectra, and you release the method. So it actually guides you step by step to build methods and validate them. And actually the daily routine analysis actually gives you the measurements in milligrams or milliliters, so the operators, the auditors, and the developers know exactly what the content you're seeing. Like I said, the software has 3 different user levels. So you have admin, developer, operator. It's got an inbuilt performance test module with polystyrene, which is recommended by ASTM. It's got complete audit trail tracking for data and use. And it's based on a client-server software platform, so you have total data integrity. It's also got features like electronic signatures, which are nowadays mandatory. I'm going to touch on a quick example of what we've done for blend uniformity. So we actually made a model on a calcium carbonate in antacid standard using this technology. So the calcium carbonate was varying in content from 32% roughly to about 50% in these antacid pills. So we took a whole bunch of calcium carbonate varying batches of these blends and did a manual titration to confirm that these were the actual values. We took Raman spectra, so in blue at the bottom you can see a pure 99.99% calcium carbonate spectra, and you can see some of those peaks overlapping in the blend itself. So this is the exploratory work that you would do to see which peaks are coming from my key active ingredient or excipient material of interest. And then you would actually build a method in BWIQ PLS regression model. And you can see we got a very, very nice uniform PLS1 model with R-squared values of pretty high, about 0.98. And several validation spectra were also collected, which are shown in red. We did 5 independent prediction samples, and we got extremely accurate readings. So this is just a quick example of what you can do in terms of assessing blend uniformity in your samples. It's pretty much all I have for the talk, but we're in booth 1612 if you want to get a quick look at the system as well. So feel free. Thank you.